Okay.
Do we have sound and do we see the...
We should have audio now online.
Should be okay.
But there wasn't for that person.
I don't mind.
We're just going to wait 2 minutes, Marion, before we start, so you can set up the presentation, so that we are on time for the online, if people are online.
Yeah. That's whatever online. So.
It's all good.
We just want to wait until the time, because if people are coming online to listen to a specific student, it's better that we don't start before.
Okay.
We're a bit early. Alright, Mario, if you want to set up. And whenever you are ready.
Just setting the time. Ooh. Okay.
You can also use the swipe. If it's connected, you can also swipe on the screen. If it's connected.
Yeah, I'm just going to stick with the
Fine.
keyboard.
Fine.
Good afternoon, everyone. Thank you for coming. My name is Marion Billy, and today I will be presenting my thesis titled Scale-Dependent Spatial Patterns of Biodiversity in the Western Mediterranean, an Indicator-Based Approach. First, a little bit of background information. The Mediterranean Sea is known as a biodiversity hotspot, but faces currently significant anthropogenic pressures. These pressures are then leading to changes in the state and dynamics of marine ecosystems. Moreover, these pressures are acting at different spatial scales, meaning that, for example, overfishings tend to have more to be like a more localized issue, whereas climate change have more global and widespread impacts. For these reasons, there is an urgent need to identify biodiversity changes, quantify them, and understand the influence of spatial scale. In this context, I wanted to take part in filling this gap by assessing the spatial variability of demersal communities' biodiversity using complementary indicators in a multi-scale approach. So I'm going to break down this in three main objectives. First, to explore the differences between shelf and slope communities. Second, to use a variety of indicators, which are taxonomic, functional, and resilience, to capture different aspects of biodiversity and evaluate the differences we can have corresponding to different indicators types. And third, to examine biodiversity patterns at 4 different spatial scales, from fine to large, and evaluate the scale dependency of indicators, which means how sensitive they are depending on the scale. Now moving to the method, I think you heard a bit about this in Sci's presentation. I'm sorry if it's a bit redundant. The Western Mediterranean is a very heterogeneous area with strong gradients of temperature, salinity, and primary production. And this is contributing to create a diverse range of habitats that are supporting different communities. Here we decided to split the communities in two, the shelf community and the slope community. I use species abundance data from the Mediterranean International Trout Survey, also called MEDITS, which covers the period from 1999 to 2021, and is coordinated across the whole West Med. So this is a super valuable program for us because it's a large and consistent data set. We selected the 233 most prevalent species that includes demersal fish, decapods, and cephalopods. And this resulted in 7,416 holes in the shelf, in the blue, and 5,248 holes in the slope. Then I aggregated these holes that were dots per year in a single value per each spatial unit within each spatial scale. So we have the first spatial scale, the finest one, which are hexagonal grid cells of 0.1 degree. Then sub-regional ecoregion based on existing knowledge. Fisheries management units, GSAs, that defined by the General Coalition of Fisheries of the Mediterranean. And the largest one, connectivity-based provinces. Here are the 13 indicators I used in this study that are divided in 4 categories. We have the taxonomic ones that are usually used in biodiversity assessments. I think you all know species richness. And while they are very useful, they do not provide a complete picture of biodiversity, so that's why I chose to use less common types of indicators, such as functional and multidimensional functional indicators. And as you can see, they are not only computed with species abundance data, but also traits data. I will come to that in a minute. And we also have the resilience indicators type. And if you need any precision on any indicator, please ask me in the question because I don't have the time to detail all of them. So as I've just said, for trace data, for multidimensional functional indicators, I use traits data, 5 categorical traits, and this set of traits with the number of categories resulted to 144 combinations that we are going to call functional entities in the rest of the presentation. And these functional entities allow to account for the functional role of the species and reduce their number, based on the fact that the species that are sharing the same traits are thus falling in the same group and are performing a similar functional role. Now that this is set, I want to introduce some general thoughts about the results I have. The shelf species were grouped into 53 functional entities, and the slope species were grouped in 54 functional entities. We had some correlation between indicators. We were expecting this kind of correlation because of the way the indicators are computed. The indicators that are depicting different aspects of richness have a positive correlation, as well as the Shannon and Pielou indices and the 2 ways of calculating the portfolio effect. We also found a negative correlation between portfolio effect and synchrony. Then we calculated the coefficient of variation for each indicator in both communities within each spatial scale. And I found a consistent pattern between indicators types where resilience indicators showed always higher percentage of variation than taxonomic indicators, themselves always higher than functional indicators. Between communities, there was a similar level of variation, except for some indicators where the shelf community showed higher variation. And there was no consistent scale with higher variation for a given indicator type common to both communities. Regarding the spatial variability, there was a very large spatial variability across all indicators at all spatial scales, but no clear gradients or patterns. This is the plot summarizing the main aspects of my results. This is a lot. You have the 4 categories depicted here with the names of the indicators in columns, and the rows are depicting the scale, so from B to D, fine to large. And there is no the scale A because it was too much spatial units, more than 500, so it was not possible to plot in this kind of plot. For a general description, I'm going to take this example. You have the color codes for the communities. the spatial units of the corresponding scale. Here it's scale B, we have 15 spatial units. And this is the axis depicting indicating the values of the indicators with the minimum and the maximum for all the scales combined. So for example, here it's species richness, and the maximum is only reached at scale D. Regarding the differences between communities, we found that in Shelf community there was larger values in species richness and functional indicators, with the differences being more pronounced at the scale C, the intermediate scale. In the slope community, however, the larger values were found in the other taxonomic indicators, functional entity richness, multidimensional functional indicators, and the portfolio effects computed with the CD method. Between the communities in resilience indicators, the values were similar, but they were not, but depending on the spatial units, sorry. Depending on the area. Another major goal of the study was to look at the cross-scale effect on the indicators. So now let's focus on these bar plots, who show the differences between the scale C, the GSAs, and the scale B that are nested within the GSAs. So each bar here is representing a subregion, with the dark and light colors respectively corresponding to shelf and slope communities. Here I framed the indicators that were the most consistently influenced by the change of the scale. And for all of them, this indicates a higher biodiversity with larger scale. However, other indicators were influenced in both directions, depending on the area and also depending on the community. Another interesting thing about these plots is that there were 3 areas that stand out, and they were most affected by the change of scale. The Gulf of Beta here, Sicily, and the North Catalan Sea. So I framed here the corresponding colors. So if you see that the Gulf of Beta here is very high all the time, meaning that the increasing scale is changing a lot their values. Moving to the discussion, there is a high heterogeneity and discontinuity across demersal communities, across geographical areas, and across spatial scales. There were very different responses depending on the type of indicators, which means that functional and resilience indicators are providing additional information. Regarding spatial scale dependency, the indicators are suggesting higher biodiversity at larger spatial scale due to greater habitats and species variety. And also, larger scales are likely to buffer against disturbances via higher functional redundancy and diversity. The indicator traits are likely to vary by the delineation of the spatial scale components, which are the boundaries, the spatial extent, and the spatial grain. The broad scale indicators produce more generalized trends, while the smaller scale reveal more specific local variations and hotspots of abundances. And as a scenario, we have propagation, masking, or divergence of a local scale with larger scale. As an example, I can talk about the Catalan Sea and the Ebro Delta that are neighbor, but very distinct, and they do have a coherent spatial structure within them. But when we move to a larger scale, such as GSA 6, we are then masking their local trends and just averaging them. As a conclusion, this study was aiming to provide a comprehensive assessment of biodiversity in the Western Mediterranean. The importance of using multiple indicators and considering scale effects in biodiversity studies was highlighted. And the findings support scale-aware management approaches for marine conservation for more tailored and informed decisions. I would like to thank everyone that helped me and contributed to this thesis, and thank you very much for your attention. much.
You rushed, I guess. You had a lot of time left. We can come back to you after if it's for a question. I want to give opportunity to members of the jury first. Yeah? What?
It's the other room different.
Nico, you want to go first?
Sure.
Alright. Thank you.
Well, Mary, very well done.
Yeah.
Nice presentation. I really enjoyed it. I absolutely like how you address the public here and how you point to the different results in your slides. That was very good. You didn't hesitate. Very good presentation. I like it.
Thank you.
I see. I didn't understand very well. Well, first, in your introduction,
Yeah.
I know that this is the part that you can explain things more broadly and don't get into detail. But do you think really that the effects of climate change do not vary in a small spatial scales? That climate change affects the whole sea or the whole marine ecosystems in the same homogeneous way through many different spatial scales? Do you think that's true?
I do not think that it's homogeneous, but I think that looking at 0.1 grid cell is maybe not the adapted scale for looking to that, and it makes more sense to look at regions. And so it's just about tailor depending on the goal of the biodiversity assessment and what we want to look at, which scale is the most appropriate.
Yeah. I'm asking because you said that the effects of fisheries are more localized than the effects of climate change. Well, sometimes it could be the opposite. Sometimes because of climate change you have cooling, then warming, and then cooling in a very in the mesoscale or even in the smaller spatial scales, and the opposite. Sometimes the effects of fisheries affect whole basins. So it's not always like that, no? It's just a little thing.
Yeah, I was thinking in the Mediterranean, since there is like the clear gradient. Yeah, I was applying this to this area, but maybe you think of something specific. I don't know, and but yeah, it's true that maybe I don't have to generalize so much, and it's just about adapting to Thank you.
Going more into your thesis, can you explain a little bit more what you mean by scale in your analysis? Is it scale B or C? Well, what is? I guess it's a spatial scale, right?
Yeah.
But as it is not translated to kilometers or.
Sorry.
I skipped this. I didn't understand it.
Okay.
You can go through it again, please.
Okay, yeah. Sorry, maybe it was not clear enough. I numbered the scale with A, B, C, D, and for the radar plots, all the plots together, I was looking at these three scales, and for the cross-scale effects, I looked only at these two ones because these spatial units are nested within these ones. Well, this one, it's very difficult to compare. So I focus only on the B and C one.
Okay.
And do you want me to explain again all the scales?
No. I think it's fine. I got it now. Although I missed that you could link these four scales with some objective measurement of distance or surface. You know what I mean? So you can link this with processes, oceanographic processes that operate at different scales.
Like, you mean like the hierarchical
you could say for
processes
example, you could say for example, final scale grid cells are processes or are patterns that occur in the scale of hundreds of meters, something like that.
Yeah.
That would it would have
maybe
been
it's
much
more
clearer
intuitive
to me. Yes,
yeah.
At least to me, yes.
Okay, thank you for this insight.
And that's it. Very good presentation. Congratulations.
Thank you.
Thank you, Nico. Karim or Rodrigue? Karim?
Yeah, actually, could you explain what connectivity-based provinces is? Because I have the doubt about that.
This is about how larvae are connected. It's like regions I took from a paper. If you want to see the references, it's Berlin et al. 2014. And it was to take a larger scale that made sense in our case. So I was thinking of this one. Maybe it's can
But
discuss
is
this.
there troll data from the middle in?
Yeah, no, of course it's true that it's like I could have cropped because of course the our dots or holes are only on the coastlines here. So maybe to avoid the biased interpretation, I could have cropped the middle part.
So since this is troll data, fisheries troll data, I assume that like rocky bottom is underrepresented. Hard structure bottom is underrepresented. What do you think that would imply for your, if you had data on rocky bottom as well, would you expect very different results? Or?
Yeah, I don't know in which sense, but definitely it would affect the results. Yeah.
No, because I was just thinking, because at the very fine scale, it could be that they could trawl over rocky bottom, or maybe there's.
It's
or
from
let's say
50 meters. So
I'm just trying to think that maybe there's different habitats are more or less represented depending on the scale that you use for the sampling.
Yeah, good point. Thank you.
Madame Poulsen.
No, I'm.
Adrian?
What's your response variable? What's biodiversity?
Sorry?
What is biodiversity?
In my point of view? What do you
In your
mean?
study, I mean, it's your response variable, right?
I don't have any response variable.
No.
It's just like visual inspection, and it's like number of individuals per kilometer square corrected by the swept area by report.
So you are using, I mean, in your title, biodiversity,
Yeah.
basically means species richness,
Species abundance is the data we used, and then we look at the functional roles of also traits or some indicators. Maybe it's clearer.
So some of your indicators are diversity indices, and you're using them as indicators of spatial patterns in biodiversity.
I compute each indicator in each spatial unit. So I have basically a value for each spatial unit. I can show you the maps I put here.
Yeah.
Like I don't know if I can zoom somehow. So I have so many maps, but like basically, you know, like for example, it's species richness here, and there is a value in each spatial unit for the four spatial scale in shelf and slope.
So are these diversity? Are these biodiversity?
This is an indicator of biodiversity. We want to quantify the biodiversity, so we were like, okay, we are going to quantify the biodiversity, trying to capture different aspects of it with 4 categories that we built.
Okay. So these are different ways that you would use to define biodiversity. So in which way is the Mediterranean a biodiversity hotspot?
Because they are it's very diverse. There are many endemic species.
More so than the Indo-Pacific?
It's also relatively to its size. It's a quite small sea, and there are like very much species, diverse species.
Are there? I don't know.
I don't like its literature review. I haven't like made it fish.
Okay. But you're basing this only on fish, right?
Yeah, demersal fish, decapods, and cephalopods.
Okay.
233 species. The most prevalent one in the area.
Can I ask you for one more definition? Because I'm not sure what you mean. What's resilience?
The resilience, in all case, as a general concept, is the ability of an ecosystem to recover or resist to disturbances. And we use 2 ways of defining it. The synchrony of the species fluctuations. So if they're all synchronous, then they have less resilience. And also the portfolio effect, it's a stability index.
Whether that is,
You're not convinced?
No, it's rather a new way of looking but I'm not sure whether biodiversity people think that it's useful in other contexts other than long-term changes.
It's also all the activities that depend on these ecosystems.
Okay.
Yeah.
Yeah. So going back to this question of resilience, the studies have shown that the higher the biodiversity, the more resilient the community is. So based on your analysis of this area, would you say there are some parts of the Western Mediterranean that would potentially be more resilient to change, or?
I was expecting to see more clear patterns of this, and I've seen that there is no high values of each indicator in one area. It seems like there are trade-offs between indicators and that we cannot achieve a high value for every single indicator. So yeah, my hypothesis was that there was maybe some gradients of or areas standing out, and it's not really the case, at least so far. From what I analyzed, maybe it needs more analysis.
It would be interesting to more or less superimpose on these figures you have a map of the fishing effort.
Yeah.
To see if there's any correlation between.
I was looking recently at the human pressure index and thinking of maybe superimposing this and connecting everything together. But
Can I ask one more question? On your almost next to the last slide, you have your conclusions or the
Yeah.
final scale dependency.
Yeah.
Next. So, I mean, were you surprised by any of these? When you spoke, you sounded surprised.
No, of course it's expected that there is a higher diversity with larger scale due to diverse habitats, but it's to highlight the hierarchical structure of the spatial processes, and that's, larger spatial scales is providing higher biodiversity, but it's not the case in all our indicators, so it's not that evident.
Do you think that the masking effects that you see mean that we should really concentrate on what on local processes?
Depending on the scope of the.
No? Audrey?
Yeah,
Yes,
so.
please?
I wanted to know a bit more about the indicators that you mentioned during the presentations. Would that be possible?
Yeah. So I put it here like a large table with all my indicators. I don't know which one I can detail. Maybe I don't have the time to do them all. Like. Which one?
Is your favorite?
Your favorite?
I think maybe the difference between functional indicators and multidimensional functional indicators can be a bit tricky and not intuitive. Basically, functional indicators are based on how the species are distributed across the functional entities. So, for example, here we have the 4 functional indicators represented in this graph. We have the functional entity richness, which is basically as the species richness, we just count the number of functional entities in a spatial unit in an assemblage. The functional redundancy is the average number of species in the region. However, this indicator can be a bit tricky since the distribution can be skewed or even. So to account for that, there is another indicator called functional over-redundancy, and we calculate how many functional entities are over this mean average, this average of species. I don't know if you're asking. And the functional vulnerability indicators is the number of functional entities with only one species within them. So if the species disappear, then basically there is the functional role is not performed anymore. And yeah.
There was another question? No? No? Yes?
I'm interested, I mean, maybe it's not on your study, but I'm interested because I'm from there. Do you how they divided the ecoregions of between the North Catalan Sea and the
In the Ebro Delta?
Yeah.
Yeah, it's like on ecosystem, on ecological reasons. Let me check this one. So, for example, this one, fisheries management units, it's like for making the fisheries convenient. It's not really based on ecological reasons, scientific reasons. While this one is based on literature and like the characteristic of the habitats, what is happening there. So the Ebro Delta, for example, there is the runoff of the Ebro, and there is it's almost only shelf. There is like the slope. It's very shallow compared to the North Catalan Sea.
All right, we have to wrap it up now. Thank you very much, Mario. We call our last presenter of this session, this stand, so it's Leah.
Come on, I put it in 4 different. Do you have your presentation on your computer?
Yes.
Because the file I'm trying to download from it doesn't work. I can't even see it on Mac.
Okay. I have a Mac as well.
Oh, that makes it even weirder, but I tried to make a PDF, a PPT instead of a PPT, and then none of them are appearing here. So they're on the stick if I'm on my computer?
But you don't see them.
Where are they? They're not.
They're just not here.
Maybe that's they are.
Yeah, room 4, session 3.
But I have it right here.
Yep. Okay.
Can you put it on a USB?
Because there's the other. So I use this. Okay.
No, we have to transfer it here, otherwise it's not broadcast.
Oh. Okay.
Can you save it on the...
I'll do the big one. I cannot.
Bah, un peu, mais...
Oh, no, wait. That was the one I uploaded, so I cannot. Oh, if I can try to do what can I do?
You can't drag it there? Is it full? Is it full? No.
I don't, there is just, do you see that it says, where is that? If I copy paste.
Wait, another USB
No,
drive?
You think it's because it's full?
But there must be something wrong.
I'm saving my presentation again and
I will check for another.
PPT is it good or no? PowerPoint presentation, it's supposed to.
I don't think it's a format.
Yeah, that's what it is under. What? I have no, I have PPTX, but I cannot save it on there.
Yeah, you want to send it to someone and we have
Yeah. Do you think PDF it's?
Or can you airdrop it to the other Mac?
AirDrop, this is true. But this is not my Mac.
I think it's easier to save it as a different format. So you can just put it in the
As a PDF?
yeah. If you don't want animations, that's the best.
They're suggesting under a PDF, like as a PDF.
If you can drag it onto the drive.
Saving it again as a PDF.
Do you have any move stuff in there?
There is a short, yeah. Maybe that's why.
Then it's not a PDF.
It's not gonna be there in
Um,
the PDF.
Wait, but it's in an additional slide that it's fine if I don't show. PDF. No.
No,
cannot.
so it's the drive.
They suggest this AirDrop.
Well, I was gonna say, can you wireless? But I have no idea about Max. If you can wirelessly transfer it.
No, because it has to be on that Mac. Can I touch this Mac?
I have no idea about Max.
I need to AirDrop the mouse.
Is this the mouse?
Can you pause the streaming? Because now we're on.
AirDrop is on already.
Can you send it to AirDrop? Do you know how to? That is this one. Partager, yeah. Oh my. I'll do it as an AirDrop. Do you know what's the name of this one?
No, but we'll see it. Just see what comes up. Octopus is Mac. Well, that's helpful.
Yeah, that's mine.
Oh, that's yours.
No, iPhone 2. Like Octopus is Mac is this one.
Oh, yes, here.
Yeah. Except.
It's there.
Great.
Okay,
Options, open.
Okay.
So sorry about that.
No worries. It's okay.
Let me just double check if the streaming is still okay.
Thank you.
Should be good. Sorry again. Good luck.
No worries. Maybe it wasn't my mistake. I don't know.
Tim, can you please close the door? As we'll be finishing maybe after the others.
Okay.
Welcome, Leah, and whenever you are ready.
Okay, thank you. Hi everyone, my name is Leah and I'm going to present you my thesis that I've done at CCMR in Faro, and it's about the effect of ocean warming on the embryonic development of Scyliorinus canicula, also called the small spotted cat shark. So the ocean is absorbing most of Earth's excess heat, so the ocean is warming, which is impacting marine life in many different ways, including in Lasmo branch. And these species are already threatened by a lot of different pressure, anthropogenic pressure. So the impact of ocean warming on these species could be even greater or more important. And so it's especially for oviparous elasmo branch, so the one that do eggs, and especially for the embryos, because the females, they lay eggs and they would attach the eggs on the substrates, and the embryo has to stay there until hatching. So if ever the water around is polluted or just not favorable for the development, well, the embryo cannot do anything, it cannot escape, it has to stay there. So the effect of ocean warming on the embryos could be even greater for those species, but there are not that many studies about that. So under that context, the primary and main objective of my study was to assess the impact of ocean warming on the embryonic development of oviparous shark. And we've done that by assessing and studying many different parameters. And we've done that by working with species, the Scyliorinus canicula, so the small spotted cat shark, that is really common to work with in a lab because it's easy to keep it in captivity. It's a benthic oviparous shark, and that is not threatened, considered as threatened by extinction. So also for our species on orchids, it was really interesting to work with it because, as you can see on the picture here, it's easy to see the embryo through the egg case without disturbing it. So that was perfect for us. And also there is an existing description of the development of the embryos that is done by Musa et al. In 2018, and that describes the development in 7 different stages. And we based our study, or we used this description for our study. There are other studies with this species working on the embryos, looking at the effect of the temperature on the freezing behavior. So what is the freezing behavior? Maybe everybody doesn't know. It's just the fact that some embryos, when they feel like there might be predators around, they would just stop moving and freeze for a few seconds just to hide because they cannot do anything else. And it appears that, so some studies looked at the effect of the temperature on that behavior, and it seems that increasing temperature decrease the duration of the freezing behavior, but it cannot be linked to a change in the metabolic rate. So it has to be due to another mechanism. And we wonder if it was due to change in the central nervous system. And since for the teleosts, so the teleosts are the bonyfish, so not the sharks. For teleosts and for mammals, cerebellum is involved in the fear response and also in a lot of different mechanisms, but also in the fear response. So we hypothesized that changes in the central nervous system could be seen on the cerebellum, or in other words, that changes on the freezing behavior could be due to changes on the cerebellum. So that's leading to the second objective, which is investigating the impact of temperature on the central nervous system of oviparous shark by working on the cerebellum. So to achieve our different objective, what did we do? So in the lab we had adults, and as soon as an egg was laid, we would take it and we would incubate in one of the 3 tanks that you can see here. And there were 3 different temperatures. So 15 degrees representing the normal temperature, the good temperature for them. And then 17 and 20 degrees being the more elevated temperature. 17 degrees, we took that temperature because in other studies they use it, but also we are expecting to reach that temperature by the end of the century. And 20 degrees, it's under the worst case scenario, we would reach that temperature. And so as soon as an egg was laid, we would place it in one of the tank and follow closely the embryonic development. And at hatching, we would also collect more data. But some embryos, we kept them, and we decided to put them in also one of the 3 water temperature, because we had the hypothesis that maybe they could recover. So imagine that in nature, they were developing in higher temperature, but then when they hatch, they could reach colder water. Well, we were thinking maybe they could recover, and we want to investigate that. So this is why we have 5 post-hatching treatments. They are here, representing here. And the first is just the embryonic treatment, and the second is the post-hatching treatment. So we kept them for 2 months. So to assess the viability of our embryo during the embryonic development, we looked at the mortality. To assess the embryonic development, we looked at the developmental time, the yolk consumption rates, and the emergence of the freezing behavior. So the age they were when they started doing freezing behavior. And then at hatching, to look at the effects of the effect of temperature on the health of the animals, we looked at the Fulton condition factor, the hepatosemantic index, and then Fulton condition factor, sorry, it's a factor that informs or tells us about the health of the animals. And then to see the effects of the temperature on the central nervous system, and so as I explained, the cerebellum, we looked at the cerebellum cell population, so the proportion of glial cells and neuronal cells. And then to assess if there were any signs of recovery, we kept the 2-month-old juveniles and we looked at the same parameters, and we also just added the ventilation rate. So the results for the embryo. It seems that mortality was clearly increasing with temperature, as you can see on the graph. Those are the values. And it seems that the increase of mortality with higher temperature is mainly explained by an increase of mortality during the stage 4 development. Because as soon as an embryo died, we looked at which developmental stage it was, and so we obtained this graph. Maybe an explanation would be that, well, like, as you can see, there's a lot of dark green, and so maybe we had more mortality at that developmental stage because it was the longest developmental stage, sorry. Then concerning the developmental time, overall, it's clear that animals incubated in higher temperature, no matter if it's 17 or 20, they were developing way faster than at 15 degrees, and there is not much difference between 17 and 20 degrees. And then we also looked at the total developmental time, the duration of each developmental stage, And we couldn't do any statistical analysis after stage 4 because of high mortality. We didn't have enough replicates. So this is what we could do. And we only had significant results for stage 1 and stage 4. No significant results or difference for stage 2 and stage 3. And here again, there is no difference between 17 and 20 degrees, but there is a difference with 15 degrees in yellow. So overall, they just develop way faster and everything goes faster in higher temperature. And then concerning the freezing behavior, as I said, we're looking at how old they were in days when they started doing the freezing behavior. 0 here being the day they were late. And in this study, it's the first time that we looked at, or that we described 3 different variations of the freezing behavior. Hasn't been done before. And behavior 1 is always the first one that was displayed by the embryo, and then it was behavior 2 and behavior 3 later in their embryonic development. Behavior 1 was an answer of shaking the egg gently underwater, and the embryo would just stop moving for a few seconds. Then behavior 2, it's also shaking the egg gently, of course, and the embryo wouldn't move for at least 10 seconds. And then behavior 3, it was in response of vibration, and it wouldn't move for at least 10 seconds as well. And so here are the results, and we also see that they start displaying the freezing behavior earlier when they are incubated in higher temperature. This could be signs of changes in the central nervous system and then in the cerebellum, but those are hypotheses. And that could be, And then, so here are the results for the animals at hatching. For the Fulton's cognition factor and the hepatosemantic index, we didn't have any significant results. So it seems that the higher temperature don't affect the health of our newborns. However, when we looked at the cerebellum to assess impact of temperature on the central nervous system, we clearly see that animals incubated at higher temperature, so 17 and 20 degrees, had a significantly less total number of cells in their cerebellum. So it's here, and that can be explained by the decrease of neurons, because if you see in white, which are the glial cells, the non-neuronal cells, they remain the same all the time, but there is a decrease with the total number of neurons. So, and also there is no difference between 17 and 20 degrees. So that could be the reason why we observe differences on the freezing behavior. Once again, those are just hypotheses. And this could be also explained by the decrease of the embryonic developmental time. So they develop faster, maybe this is why they didn't have less neurons, we don't know. And then for the juveniles, the 2-month-old juveniles, as I said, we looked at the same parameters. Regarding the mortality, we only obtained mortality for animals that we kept at 17 degrees all the time. The mortality was of 45.5%. And we didn't have any mortality at 20 degrees. And we think it could be due to the high mortality during the embryonic development. As I showed, I think it was the first graph, the highest mortality was at 20 degrees. And so maybe the selection is done faster at 20 degrees than at 17. And also maybe animals that hatch at 20 degrees are already adapted to live at those temperatures, but when they hatch at 17 degrees, maybe all of them couldn't survive, but if they move at 15 degrees after, they're fine. But if they are kept at 17 degrees, they are not, at least for 2 months. And then for the Fulton cognition factor, hepatosomatic index, and the ventilation rate, here also we didn't have any significant results, which makes us think that there was no impact of the temperature on their health, or at least for the one that survived and hatched. And to look at signs of recovery, we wonder if animals that were first in higher temperature and then moved at 15 degrees could recover and have signs of recovery, but the answer is no. As you can see, here all the animals that were firstly placed at 20 or 17 degrees, even though they were back after at 15 degrees, well, they still had way less total number of cells, explained by the fewer neurons in their brain. So at least 2 months after hatching, they can recover. So here are the main messages of this research. We wanted to assess the impact of ocean warming on the embryonic development, and we wanted to see if there was effects of the temperature. The answer is yes, because clearly we had an increase in mortality, we had a shorter embryonic development, and also earlier display of the freezing behavior. And then secondly, we wanted to see if higher temperature were affecting the central nervous system of animals, and by looking at cerebellum, and the answer is yes as well, because there is a clear decrease of the total number of cells and number of neurons in their cerebellum. And this change seems to be irreversible, or at least for 2 months. And so these results are really interesting, and we believe that further work should be done, because once again, there is not much research about that topic. And we think we could do it again, but with more replicate, because due to high mortality, there were several times we couldn't do any statistical analysis because we didn't have enough replicates. Also, if we keep the embryos for more than 2 months after hatching, we might have different results, because maybe after 3 months we would have more mortality, or even ventilation rate would change, or other things. Because sometimes some of the results that I didn't show, the graph that I didn't show that were not significant, sometimes they showed tendencies, but nothing was significant, so we cannot draw a conclusion. Also, maybe doing or adding a study of the freezing behavior and the duration of the freezing behavior, like the other studies did, but we didn't. That could be interesting. And also see if there is an impact of the change in the central nervous system. So if the decrease of neurons in the cerebellum have an impact on the animals when they hatch, doing, for example, behavioral studies, because, as I said, for other animals like teleosts and mammals, the cerebellum is involved in a lot of different parts of their behavior and learning and coordination, for example. And we think also something that could be interesting is to simulate heat waves. So by placing the eggs in higher temperature, but for a shorter amount of time, because that would also happen in the future. It's also already happening now. And that could be interesting to know what are the effects of that. And so I would like to thank my supervisor and thanks the students who helped me collecting the data and the lab technician of CCMR. And thank you all for listening.
You were precisely on time, so I'll commend you on that. So I'll ask the members of the jury to start the discussion. Who wants to go first? Won't fight?
I'll do it.
Okay, thank you, Lika.
Okay, Lea. Yeah, very well done. Very nice presentation. I really enjoyed it. I like the combination of photos and figures. Although at these last slides, too much text.
Too much? Okay.
Yeah. I would like, you could put this in a diagram or something like that to make it easy for us to grasp your take-home message on the discussion, for example. But overall, it was a very good presentation. I really enjoyed how you addressed the public and the jury here, and how you were pointing at the different results. All that was very good. And about the content of what you did, I have 3 main questions.
Yeah.
First one is, why those temperatures?
Why? Because that's what is predicted by IPCC. So by the end of the century, they're expecting 17 degrees. They're expecting to reach 17 degrees, because they're expecting to have +1 or +2 degrees of the temperature of that depth. And
at that depth?
At like where it's 15 right now. At those, because I didn't say. The eggs are not laid super deep. They are laid in higher layers of the water. Okay. And so the adults and the juvenile, they live deeper in the water. Well, not like, it depends on the location. But and the eggs are always laid in higher parts of the upper layers of the ocean. So 17 and 20 degrees. Also, it's because the few studies that we read and that were assessing impacts of increasing temperature on oviparous sharks were using those temperature. Sometimes they were only using 15 and 20, and some they never went higher than 20, and I never read or something else. So 15, 17, and 20, and it's because
The APCC says that.
Yeah.
But do you know exactly the scenario or?
But that's what I said. Under the most probable would be at 70 degrees. That's what we're gonna reach at the end of the century. And under the worst case scenario, that would be 20 degrees.
Looks kind of cold to me.
Cold?
Yeah, I was expecting more.
Because 15 is the normal temperature. Plus 5
15
degrees
is the normal temperature.
for them to for them to develop.
And did you take into account the seasonality of temperature?
No, that we didn't. I know. Yeah, one study was doing it.
Maybe
But we didn't.
Maybe it's warmer at that season in particular.
Yeah, that we yeah.
In the spring, summer, I guess, no?
Yes.
The other question is that you have some data of the amount or the number of nervous cells there in the cerebellum.
Yes.
And you have that result on one hand.
Yeah.
And on the other hand.
For two.
Yeah. And on the other hand, you have your result on the percentage of behaviors that are shown. Behavior 1, behavior 2, and behavior 3, right?
Yeah. Yeah.
Why
freezing behavior.
The freezing behavior, yes. Why not correlating them?
Because we're not sure. It's just a hypothesis that they're linked to that. But we don't know. We don't even know anything about the cerebellum of sharks, so we don't even know if they're linked to fear response. Yeah. And we use the cerebellum because, as I said, for the mammals and teleos, that's what's involved in the freeze response. But yeah, you think
you don't have to be sure. You are just guessing here a little bit.
Okay.
If there is, I am asking you to do a correlation. Correlation doesn't mean causation. Doesn't mean that is the cause, but it's an indication that it might be the cause.
Okay.
So you can do a correlation. Next, you put the number of cells that you are counting, and in Y you put the percentage of behavior 1, for example.
But those are the age at which they started displaying the freezing behavior in the 3 different temperature.
But don't you have? I think you do have the behavior 1, for example, the percentage of animals that exhibit behavior 1 at one given point, right? Or not?
At one given, well, it's in days. Like I don't know by heart the number, but like here you can say that on average all the animals in 15 degrees were starting freezing behavior 1 around 58 days old. Yeah. But it changed for each individual, and it changes in each treatment.
Yeah, but in other words, can you associate each of these bars with a mean value of number of cells in the cerebellum?
Yeah, I could. Yeah.
So you can make a correlation.
Okay. Yeah.
And that would make a lot of sense, and that would reinforce the idea that it might be a relationship.
Okay.
You don't have to be sure about it. You just have to try.
Okay. Yeah.
Yeah. I have more questions, but I think it's okay. Congratulations.
Going back to this temperature question. So these were cat sharks from Portugal, from the Algarve.
Yeah, exactly.
Okay, so what you're saying is that 15 degrees Celsius is the optimal temperature for
the embryonic development. But adults, they live deeper.
Yes, I know that. Yeah. And deeper means like 13 degrees or something like that. But this is a species that has a very wide latitudinal range. It goes from like Mauritania all the way up to Norway.
Yeah.
So surely, I mean, if you repeated this experiment with cat sharks from off Bergen or somewhere, would you expect the same results, or they would have a different optimal temperature probably?
Well, the study that has done exactly on the same species was also using 15 as the optimal temperature. However, we got different results at some points, which were for changes in the Fulton condition. So for us, there was no difference in the Fulton condition factor, but for them, they had significant results. And we hypothesized that it's because those animals were used to more colder water, and since we were moving them at 20 degrees, which was a bigger shift or bigger change, they couldn't cope with that as well as the cat shark from Portugal. So maybe the different populations are not used to the same temperature and don't respond the same way. So maybe for the freezing behavior, yes, the result would be a bit different too.
So
I
I
guess
think because there are different populations or subpopulations,
Yeah.
there is this possibility of
Yeah.
adaptability
Of
or
course.
changes in the range.
Yeah.
I mean, maybe it gets too warm for the population in the Algarve, but
Yeah.
maybe the ones from Northwest Africa might come up and find it more
And do exactly.
suitable. So it's
Yeah,
you
totally.
know, it's not such a bleak scenario, let's say, in terms
Yeah,
of
that's only for that only it's based on all results which are based on wild animals that were from Algarve.
yeah.
So yeah.
Can I follow up right
Yeah,
there?
yeah.
So, what temperature was it when you collected these eggs?
Sorry.
What temperature was it when you
15
collected?
also. The eggs or the adults?
No, when you
Collected the eggs, it was 15 because the adults were also at 15 degrees actually.
Okay, but so I didn't quite understand whether you collected the eggs from nature, or you brought the sharks in and they spawned in nature
Exactly,
in the
that's
tank,
what
and they were
happened.
not at 15.
So in the lab there was adults,
Okay.
and as soon as the eggs was laid, we would check every day if there was a new egg.
So I guess in another experimental design you would consider 15 to be your control, and your
But this
treatments are yes. By presenting it at 15, it's not the control; it's a treatment, right? Just a point
okay.
of design. But I guess I have a question about the developmental stages and developmental rate, and because you're doing a classical temperature-dependent development study, right? So do you have a slide of the different stages?
Yeah, I do.
Oh, good. I would have liked to have seen that very, very, very early. I loved your slides, but that was the one I was missing. I would have loved to see that really, really early.
So
Okay.
yeah, and there are criteria for all of them.
Perfect.
Would you like me to explain each of them?
No. I just want to make sure that there are some, and that
Yeah.
I can look at it, and
Yeah.
you can see all those things, which is great. So the duration of the stages. I mean, you would expect the stage duration to go down when you increase the temperature. You would expect time to hatching, blah blah
Yeah.
blah blah. And because the developmental rate is going up, you would expect the age at which the individuals show their behavior to come earlier.
Yeah.
And it does not have to be because of nerve damage, of
No,
neural, of
no.
cerebellum. So what makes you even think that, yeah, it's just that you've got a more advanced individual.
Because yes, it is, but no, but because in other studies they didn't look at the age at where they started. They looked at the duration of the freezing behavior, and that
But
duration.
your data.
Yeah,
your
based
data
on our
is
data.
the age at which they started
But the-
freezing, right? Of
Yeah,
freezing
yeah.
behavior.
But we were also thinking before doing our study of the freezing behavior. We looked at the other articles that were saying, increasing temperature, decreasing the duration of the freezing behavior, nothing with the age. And so there is, but it's not linked to the metabolic rates. They know that. And so because they looked at the oxygen uptake after the freezing behavior and other parameters, and so it wasn't linked to the metabolic rate. So it has to be due to something else, but we don't know why. And so because we don't know what are the mechanisms, we assume that there was another. It has to be due to something else. That could be changes in the central nervous system, just due to higher temperature. That was just a hypothesis, and
it couldn't be due to the fact that they reach that stage where they can have that behavior earlier.
Yeah, also.
And have a normal brain.
Yes, it could be, but yes, it could be.
Okay.
But not for the duration of the freezing behavior, which is not what we studied. But
no.
But then in their case, because the duration is changed, it couldn't be linked to faster embryonic development. Do you know what I mean?
But so many embryonic development studies in teleosts try to standardize the development. So how would you do that here? How could you standardize your time frame of development so that you take age and and temperature out of the equation, so you can put everything on the same axis.
At different temperatures?
Yeah. How could you directly compare the different temperatures, the stage of development at different temperatures?
I'm not sure I understand your question, I'm sorry. Scale, like, because it's visually that we see the different stages. So I would look at an egg and see, oh here, okay, today it's not in stage 2 anymore, it's in stage 3. How old is this egg? And it's not based on, they all start at 10 days, they become stage 2. And then at 20 days old, they're stage 3.
But for an individual raised at 15 degrees, their experience of time takes longer than an individual at 17 degrees or at 20 degrees, right?
Mm-hmm.
So how can you put them all on the same, how can you standardize that scale? Are you familiar with the term called degree days?
No, I don't know. Sorry.
It's like the underpinning of most temperature dependent development work with Thesis anyway.
Okay.
And,
Well, I
and
didn't.
the basis in aquaculture and
Okay.
everything. So in order to account for the differences that you see in temperature and standardize them, so you can look at everything except temperature in a way.
Okay. I didn't see that in any of the articles that really looked at or research. Like in any other articles that were doing the same kind of research, they were not using the
Degree days?
no degree dates.
Yeah.
Les degrés jour in
Day
French. Yeah. Degrés jour. in
Degree
French.
no.
Degrees per day, basically.
You add up all the time.
In agriculture also.
Yeah.
we use
Okay.
that.
It standardizes for
Okay.
temperature, so you may want to look back at
Okay.
it.
Yes.
I guess so. Can I ask one?
Very quick one. And then I have,
Okay,
and then I have,
oh,
I have a very quick one before we wrap. You talked about the insufficient replication in your study.
Yeah.
I want to ask a very general question. It might be a bit naive, it might be hard to answer. Are you happy with the experimental design? And if you had insufficient, unlimited means, what would you do differently?
Unlimited means.
Yeah, in terms of, you have all the labs, all the time, and would you do things differently and why?
But personally, was I happy? Well, not always, because you have to kill the
That's not what I'm
embryos
talking about.
have to
It's
kill.
like the design in terms of replication and things like that.
Oh yeah, I would have. Okay, yes, I would have first of all different temperature, doing different duration, coming from different adults, because also we didn't have hundreds of adults laying the eggs, so maybe there is also changes like genetic changes and individual change. I would have way many more replicates, but it takes really a lot of time to follow up each special individual, and I would do the same, but just longer period of time after hatching.
So you think the individual eggs are actual replicates? I'm not certain of that.
No, yeah,
I
that's the
think
thing.
you have pseudo-replication in this study because the unit of replication is a tank, and you have 3 tanks. So that's not
Okay.
that's not your fault,
egg
but
itself. Okay.
but I think that's a major fault in the design.
So you think for 17 degrees we had to have at least 2 tanks or 3 tanks for
At
17?
least, yes.
Okay. Okay.
Yeah,
Yeah.
because that's the basic unit of replication.
Yeah.
And you were lucky because if anything had happened to the 15-degree tank, it's
We
gone.
would have had a problem. Yeah.
Yeah.
Of course. Yes.
On top of the security, in terms of statistics, it's difficult to consider each of these eggs as a replicate
Okay.
because they're actually all linked together by the tank.
Yes. But each
Yeah.
tank. Oh yeah. Yeah.
Yes.
Each tank were individual tank. They were.
Yeah, but in the same tank you have
You have a
many.
tank with them all hanging, right?
Yeah,
Yeah.
they're all hanging in the same tank, so they're
Yeah.
linked by the tank,
Okay. Yes.
which
I see
might
what you
have
mean.
other factors influencing it than the temperature, which is the one you're looking for.
Yes.
And
But is that?
you cannot disentangle these factors.
Yeah.
That can be complicated.
Okay.
Can I push on one more?
Okay, last one, and then we'll let you
Very
go.
quick, because I think that you're interested enough to follow it through and look at it again.
Yeah.
You have very high mortalities,
Yes.
and then you have an effect. And so is that due to the mortality? Have you lost? You mentioned it a little bit, but the fact that you have so poor survival in your highest temperature
Yeah.
may affect your observations.
Yeah. Exactly.
So I think you
Because-
mentioned some of that, and Go back and look at that one again, though.
Well, for example,
Because that's very nice.
because we had too much mortality, we couldn't do a statistical analysis after stage 4. Is that what you're talking about?
No, but some of the things, at one point you say, well, perhaps what I saw here, that I don't get any differences, or I do get differences, is because you've reduced the variation between the individuals that you're looking at and using as replicates because of the high mortality. So it's something to go back and look at, because I think you would like to go back and look at your data.
Do you remember which slide?
I mean,
I
she
think it
means in
was-
general
Oh.
to look at for you.
Okay.
in
I
life.
thought she. Okay,
Not
sorry.
now.
I thought you wanted me to go on one data.
No,
first slide.
No,
Alright.
but
After,
you seem to be
next week.
You seem like somebody who's enthusiastic about your data,
Yeah.
so I'm suggesting that that's the place
Okay,
to go.
thank you.
So,
Yes.
Alright, thank you very much, Leah.
Thank you.
Thank you all for the presentations and the attention and the questions. We have a coffee break, if I'm not mistaken, now, and we start again. Sorry we're a bit late, but we had technical issues.
のだけじゃない。Oh,
I was just saying hello. That's it. So welcome back for session what, session 4 already. Yes. So in this session we'll have some Madeleine just to make sure that Faye is here.
Yes.
And Faye is fine. And already lost. I'm sorry, David. David. Okay. So your jury here will be with Nico Berg from Oviedo, Pere Arberg from Université, from Göteborg Universitet, and myself, Christophe Muquet from Université Côte d'Azur. Hello, you're really happy, you're really going to start. The Octopus is ready for you.
Thank you.
So you have the floor for 15 minutes.
Okay.
Just before you start the timer, I remind you that we have a set of microphones, so during the presentations. Thank you.
Okay, hello everyone, and welcome to my thesis titled Modeling the Distribution of Octopus vulgaris in the CS Islands. So first I'm going to start with some little background on the study area of the CS Islands, because I'm sure a lot of you don't really know where this is. So you probably don't know where Spain is, but more specifically, I was in Galicia and on the western coast here. So the CS Islands look like this, and they're an archipelago made up of 3 islands. So what's really important to note about this archipelago is that it was declared a national park in 1980, but small-scale artisanal fishing is still permitted even in the protected waters. And there are 2 main components to the island. So there's the western side here, which faces the open Atlantic Ocean, and because of this, it's exposed to really strong winds, tall waves, strong currents, and the bottom is very rocky with encrusting coralline species. And then the other side is sheltered by the land masses, so the water is much calmer and many seaweed species thrive. And then finally, the CS Islands are part of a local upwelling system, so they benefit from seasonal surges of primary productivity, which come and fertilize the waters and just promote the entire food web growth. So this diversity of habitats and the upwelling means that in this area, there's a really unusually high biodiversity. So these are just kind of some of the species we can see. But today we're going to talk about my favorite one here, Octopus vulgaris. I don't know if you spotted him. So Octopus vulgaris in Galicia is the most common cephalopod. And because of this, it's a very important commercial species. And in fact, it's one of the most fished species in the CS Islands, mostly using creels. And it's facing a lot of overfishing and other anthropogenic threats such as pollution. And recently there's been noted population decline, not just in the CS Islands, but just in the Galician region overall. A bit about their ecology. So after an initial parlarval stage, Octopus vulgaris are fully benthic and they have very soft bodies. So they're very vulnerable to predation. And because of this, they tend to spend the day in dens and they only come out to hunt. So we can say that their presence is very dependent on an adapted environment, and this makes them a very well-suited species for species distribution modeling. So what is species distribution modeling? A very basic rudimentary definition is the use of computer algorithms to predict the distribution of a species across an area of interest using environmental data. That's a lot of words, so I'm going to try to break it down for you guys. So let's imagine that we have an area of interest and a species of interest, and we want to know what the distribution of our species is in this area. So we're going to divide it into grid cells, and we're going to survey some of these cells, hopefully randomly, and we're going to find presence, so ones, in some of the cells, and absences, zeros, in others. And all the other ones won't be surveyed because of cost constraints or time constraints, or just because the area isn't surveyable, like weather and things like this. And on top of that, we're going to go and take environmental data, which we can get during the surveys and then model the rest, or we can have satellite data, there's a lot of different sources. And what we're going to do is put it into our model, which is going to take the presence-absence data and the environmental data and give us our predicted distribution. So any areas that are environmentally similar to a presence are going to have a high probability of occurrence. Any areas similar to an absence are going to have a low probability of occurrence. So, to summarize, what do we need for an SDM? We need presence-absence data, environmental data, and a model, because there are a lot of different models to choose from. So in my case, for the presence-absence data, I got it from Cephaparchus, which was a project led at the institute where I was staying in Vigo, whose goal was to describe cephalopod ecology in the CS Islands. And to do this, they did 94 visual scuba censuses from 2012 to 2014, using underwater cameras the whole time to film the benthos where they were diving. And this was the first project of its kind in CS Islands in terms of studying cephalopods. And it was a very substantial effort. As you can see here, whoa, twice. Every time there's a red dot, that's where a survey took place. So if we take the video footage that I mentioned earlier, combine it with the GPS coordinates taken during the dive, and so we look, where did we see an octopus, where didn't we? We get our first type of data, our presence-absence data, which looks like this. Next, the environmental data. So as you can imagine, there's like an infinite amount of variables that we can choose from. So we have to do an initial kind of selection. And to do this, you can base it on previous studies of the species, the known ecology of the species, so benthic and pelagic species won't be affected by the same variables. And finally, the local environment. So if you know that your area has high variability of temperature, for example, it would be interesting to take this into account. In my case, I got my first 3 variables from Cephaparchus, so that same project I mentioned earlier. They also modeled these 3 variables. So bottom substrate type, interesting to include because Octopus vulgaris is benthic, and they need dens, so the den availability won't be the same in rocky and sandy areas. Depth, because they're known to prefer shallower waters, and wave exposure, because strong currents can impede survival, so interesting to include it as well. There was another project run in the lab called the Strauss project, and from that I was able to model bottom temperature and bottom salinity layers, which are both really good proxies for upwelling, because I didn't have high resolution upwelling data, so I used temperature and salinity instead, and these are also often correlated with prey availability based on the literature. And finally, I computed variables in ArcGIS, so slope and standard deviation of slope, which come from the depth layer. These are also terrain indicators, which could play on den availability, but also upwelling, like I mentioned earlier. And finally, distance to the coast, which again, the same proxy to upwelling. Also, it was recommended in literature to include it for Octopus vulgaris. So we have presence-absence data, we have environmental data, and all we're missing now is our models. So I chose 3 types of models: generalized additive modeling, maximum entropy, and random forest. And I chose them because they were robust and well-researched, which meant that a lot of peer-reviewed papers had used them to create species distribution models, and also because they're open access, so it's a very repeatable, accessible experiment. And a little bit more about the models. So I'm going to try to stay simple, but generalized additive modeling describes probability of occurrence through nonlinear patterns by combining multiple smooth functions of the environmental variables. So for example, here, the depth. Here is probability of occurrence, so we can see that as depth increases, probability of occurrence increases, and then decreases and it increases. And here is slope, so it's the same thing on the side, probability of occurrence. And you combine multiple of these and you get your final model. Maxent is different. It's a machine learning method, and what it does is sample background locations or the available environment and contrast them against presence locations. And then it uses maximum entropy to find the distribution that's closest to uniform while integrating the limits of the environmental variables at the known presence locations. And finally, random forest uses decision trees. Decision trees classify your data into presences and absences based off environmental variables, but random forest does this by subsetting your data randomly and creating n trees. So in my case, I did 1,000, and it does this with your data, and then it takes an unknown point where all you know are the environmental variables at that point, runs them through the trees, and each tree has a final decision of 1 or 0, and then you can look at the ratio. So for example, if you do 500 trees and 400 end in 1, that's an 80% chance of occurrence or 0.8 probability of occurrence. And then you just repeat this for every point. So while you're building the models, it's also really important to look at model assumptions. So in my case, the biggest issue was GAMS and the multi-collinearity, where you have to avoid putting collinear variables into the same model. So for example, quite logically actually, depth, distance to the coast, salinity, and temperature are all highly correlated. So I never put them together in the same model. And because Maxent and random forest didn't have similar limitations as to including variables together, my GAM was chosen as the baseline. So all variables that I ended up putting into my GAM, I put the same ones into the Maxent and the RF. And finally, to assess my model performance, first I split my data into training and testing data, and then the main statistic that I used was the area under the receiving operating characteristics curve, or AUC for short. And this statistic measures how well the model can separate between classes, so in my case, presence and absence. And anything above 0.5 means that it's better than random. Anything above 0.7 is kind of what you're looking for. And performance is used to select the final model, but not only, as we're going to see in a second. So, like I said, we're testing different environmental variable combinations based off the assumptions that we have to respect. And then we're going to look at the performance statistics. So in this case, we have a bad model and then two very good models. And you might think we're going to take the one with the better AUC, but actually we're going to choose the one here on the far right because you're not losing a lot of performance, but you have one less variable, so it's much easier to interpret when it comes to the discussion. So you have to have an accurate and parsimonious model if ideal. The final variables I selected were bottom substrate type, depth, standard deviation of slope, and wave exposure, and all my steps were done using R, RStudio, and ArcGIS. And now the fun part, we get to my final predictions. So my first model, my GAM, had an AUC of 0.780. Anything that's red is a high probability of occurrence. Anything that's green is low probability of occurrence. My Maxent had an AUC of 0.878, and you can already start to see the differences between the different modeling techniques. And finally, my random forest had an AUC of 0.933. And so I have 3 different models now, and they all have an AUC that's not 1, which means they aren't perfect. And one way to improve performance is by combining your models into an ensemble model, which basically takes what's true in all your models and boosts this while reducing all the error in your models. And to make an ensemble model, we're going to look at an example for just one cell. So you have to start by multiplying your prediction in the cell times the AUC, and then you get a weighted prediction, and then you're going to sum your weighted predictions and divide them by the sum of the AUC to get a final ensemble prediction. And then hopefully you get something like this with an even better AUC, which I was lucky enough to get, so 0.948. And so this is the final model that I based all my discussion on. And if we look closer, we can see that the main points of interest are the very high probability on the eastern shore compared to the western shore. Though there are some anomalies up here, they can be explained that because they're very shallow expanses of raised rocks, so the depth is similar to this side. Anything on the western side that was high probability was very, very close to shore. And finally, this smaller islet here probably provides a similar environment to the eastern shore, which could explain why it's red. So what can we say as a general trend? My conclusion was that Octopus vulgaris prefer warm, shallow, and sheltered waters with uneven sandy bottoms. In terms of warmth and shallowness, this follows all the trends that I found in literature for Octopus vulgaris, but also just for cephalopods in this area in general. My hypothesis as to why the octopus prefer sheltered waters is that it's less straining for movement and easier to hunt in, so it just boosts their survival. I think the sandy bottom, based on some papers that I read, could be related to den availability because they are known to burrow in the sand. They also do like to go in crevices, so this part needs to be investigated further. It's also possible that their prey is mostly available on sandy bottom, so when they're spotted, they might be out hunting and on the sand. And finally, the eastern side of the island provides the ideal habitat in all of these aspects for all of these environmental variables. And so finally, I'm going to discuss some future recommendations I have. So first, I would recommend more surveys to be done presently because these are 10 years old now. And I don't know if you remember, but the map we saw is not a completely even distribution of surveys. So it would be interesting to go and survey the less studied zones as well, see if we get the same results. I think we can integrate more environmental variables. I was limited due to time, but also because it's a very small study area and there's not a lot of open access, high resolution data available, for example, for upwelling. So we can make even more accurate models. I actually had data from Cephalparcus that I couldn't even use, such as size class and life stage, which would be really interesting to model as well. And Finally, I think these could serve as a foundation for this area that, like I said, is protected but still remains heavily fished, and could be really interesting to establish no fishing zones in the population hotspots, even seasonally, just to help mitigate this population decline. So thank you so much for listening to me, and thank you again to my supervisors, Jorge and Miguel, for all their help.
Madeleine, also on your time management. We can start with you.
Well,
Start with
thank you very much for this very nice and intense presentation. I really enjoyed it. I think you made a really good job addressing the public and showing the results, especially the complex statistics that you show here. You really take your time to explain them step by step with nice examples. That's very nice. I really appreciate that. It's very good. And about the content of your presentation, yeah, I have 4 questions mainly. One of them is if you can go back to the wave exposure index, or how did you calculate that? That's just it take into account orientation and the fetch.
Oh my goodness, where is it? Here it is. So these were given to me by my supervisors because it was modeled during Tapa Parkits from someone who's much better at modeling than I am. And but I think it was based on local weather weather data, and then it wasn't just like, I mean they took like multiple versions to get this final one, but again it is just a model, so it's quite accurate because, like I said, this area is like much more violent. I mean that's why the eastern side wasn't as dived, yeah, I don't know the specifics about the modeling.
Yeah, probably it's the Meteo Galicia model that they use quite a lot in that area.
That rings a bell, yes.
Yeah. Okay. Then you didn't use upwelling as a variable itself, because I understood that your spatial resolution or for the calculation of this index wouldn't be that good.
Yeah, I had, so someone, the Strauss project gave me upwelling data, but it was one value for my entire study area. So if I look at seasonality, I could use upwelling, but I didn't have time to look at distribution through time. And also just my occurrence data, there's only like 94 surveys, and if I started dividing it by season, it was less and less accurate, or like less and less samples. So I preferred to stay with one model.
But how long is that coastal stretch? Like
this one, I know the total here is 8 hectares. Hectares, yes, but in kilometers, I'm not sure. But each pixel is 20 by 20 meters.
Yeah.
It's
Okay.
very small.
It's a pity because I did calculate myself an upwelling index for this coast. And not just for CS Islands, but for a bigger coastal stretch.
It's not finished. I have to publish still, and they want me to do more things. So.
You can do it with some high-resolution models like Ostia, for example, from UK.
Yeah.
It's called, yeah. I'm Spanish and I also laugh at it. Yes. And also MUR from NASA.
Okay.
They have a higher spatial resolution, and you can calculate. You can extract the data of sea surface temperature from the coast and sea surface temperature far offshore. And that difference, well, you made some more calculations, but that difference tells you actually if you have upwelling. If it's much colder onshore than offshore, that's upwelling. If not, you don't have it.
Yeah.
So that it's more precise
to
capture upwelling variability than just temperature and salinity. Yeah, it's published. So.
Yeah, because I know normally you also can use like chlorophyll coverage as like a very close proxy, but that was also not available to me.
It's not the same, because sometimes you have an upwelling hot spot, very cold waters there, but when the phytoplankton reacts, that's already 2 or 3 days later and in another location. So it's not the same. And in this coast, it happens because you have a strong currents there.
Maybe I will send you an email.
Yeah, I was about to propose you to. I have another question.
Yeah. Actually, a couple of them. The GAMs.
Yes.
I saw that you were showing the different shapes that the GAMs can have.
Yeah.
But you have to be careful with that because sometimes you are not sure if that shape has a biological meaning or not. Why?
In terms of the
why it rises here, then why it drops, why it rises again. It's good. The GAMs are good for for fitting this kind of data, but you must have a biological meaning of each of these curves, and that's very difficult sometimes.
Yeah, I see what you mean. Yeah.
But overall, very nice design. I really like it a lot.
Thank you.
That's not the question. Though.
Yeah.
Okay.
Yes.
Yeah, thanks for a really good presentation.
Thank you.
I wonder, you start with the data that they did there, and they actually looked at the things. So, and then you model everything and put some environmental things on this. And what's the extra you get from the model compared to the field data that they had from the beginning? What extra do you get from the model?
Having the distribution map?
Yeah.
What do your model add to the observations?
Yeah, well, you do have a lot of observations, and from that I could say that, well, the octopus will be here and there, and now you're using a model. So what extra thing do you get from the model that you don't get from the field data directly?
I think, first of all, the maps have a visual component that's really interesting when you're trying to inform legislator, because just showing points on a map is not always going to speak to people as much as having this very red side of the island. But also the surveys had varying abundance as well. So I don't know, I think it's interesting to have distribution maps, because for me it's just a much clearer image of where the octopus are. And then also because, like I said, the surveys were not completely homogeneous. Now you have areas that are red on the map where we haven't dived. So as a follow-up as well, to do more surveys and then collect extra data, it kind of gives you a jump start about where you think where you should be going.
If you make this even more general, you think you can apply this model to another area?
Yeah, so what I was...
As a predictive model.
So these types of models tend to be you don't want to go too far out of the range, but this is the CS Islands are part of the PNIA, which includes 2 other archipelagos, which have a very similar environment to the CS Islands. So I think this model could be used to apply to these areas where I also know no octopus surveys have been done at all.
Yeah, so I just took off one of my questions. Thank you, Paddy. No, I really appreciate the fact also that you mentioned, for example, that it's important to start with simple models, or at least to select a simple model, even if there is less. It was really clear, really. Maybe I can come back on the upwelling, something I didn't get at some point. You said something about how the distance to the coast could be a proxy to an upwelling, and I didn't get that.
Yeah, what I found in literature is that closer to the coast, there's more upwelling. It's not as good of a proxy as for temperature and salinity though. And I included it mostly because it was very easy to compute in ArcGIS, and I could and then I tested it to see if it was since it had a high correlation with the other 3. I kind of wanted to know which one of the 4 was the most significant for octopus presence absence, but it was a variable that I chose to include because it was easy to compute, and I saw it in other publications, but I think I knew that temperature and salinity would be better in the end.
That's upwelling, because I guess there are other reasons why they could be or not upwelling.
Yeah, but it's yeah, I wasn't expecting great things from this variable, but I did want to test it. Yeah.
If you can come back on the map of your final prediction. Yeah. Oh.
Okay.
That's your big results. You have to be proud.
No, I mean, it's been a long time.
Just before.
Oh, no, it's after this one, the final one. This one?
Oui. This is the final.
This is the ensemble.
Yes. What is the reason of the lines of the patterns, and do they mean something, you think, for
the lines here?
Yeah.
These are the lines that I observe on the slope, on the slope line. So these are like areas where it goes up or down very sharply.
Okay.
But the slope for me is the hardest one to interpret in terms of biological like significance.
Yeah.
So I don't want to like say something that's wrong, but I know that these are connected with slope.
Okay. So maybe not with consequences on the biology of the object.
Yeah, it's hard to say.
It might be simply an artifact to recognize, maybe like that.
But since it's so light green, it's not very important.
You talked about the present absence of the octopus and environmental data. It's not environmental data, but if we have a look to the food, can we consider the food of the octopus also to fuel your model? What do they eat, and can you get some information about the distribution of the food?
Prey distribution maps are not like readily available. At least I tried to look for like fish, and then also I know that my supervisors were trying to look. I mean, this is not for octopus, but like just in general, there's a lot of like work being done here, but I couldn't find anything.
This job that you're doing hasn't been done already on the food. Because
no,
food has been modeled.
They didn't do any distribution maps of anything in the CS Islands. I'm the first one.
So let's look at the future. If someone's, maybe you, found a way to model the same way the food of the octopus, so the prey distribution, how could you take that into account into this kind of model?
Into the model?
Yeah.
It's very easy. Once you have the layer with the distribution of the fish, you can just put it as a variable. You can, if you have the raster, you can just extract it in ArcGIS as a CSV, and then the CSV you put it into all your models. And
what could you imagine it could happen?
Well, I'm sure that prey abundance has a role, because the octopus, like this is anecdotal, but I know that the big like hotspots of octopus move a lot on this shore. So all of this is red, but like the fishermen, they change where they put their creels very often. So they're moving a lot on this side. So I think that the octopus probably follow the prey, like when they move. I don't know. I think this could be interesting. I'm sure like whenever you have prey distribution, you want to include it in your model because that's the biggest like relevant factor to where your species is going. But since you never have this or almost never, you always use like other proxies. Yeah.
Also, you were saying that you were trying to avoid having correlations between variables.
Yeah.
But it's also possible that the prey distribution could be also, there could be correlation between your prey population and environmental data.
Yeah. So like I said, the temperature and salinity, they're proxies for upwelling, but then upwelling is also a proxy for prey because where there's upwelling, there's everything. So it all kind of like connects, but then you get further and further away from the source. So it's always better to have the original data. Yeah.
I want to add another question. Because we have, think about it. Your hand is raised for a long time.
I have two super simple questions.
One, because we have another one here.
Okay, only one. Okay.
You can ask me after.
No, yeah. I don't know how mobile this species is, but my question would be how confident you are on the absence where you have absent, because on highly mobile species, they always do zero absence. So how confident you are?
Well, they do have some site fidelity octopus because they have their dens. It's true that, so again, depending on the age that they are, they're more or less coming back to the same area. And if they've been disturbed, they won't go back to this area. But there is some site like fidelity with their den. They always go hunting like around the den and then come back. But the absences, we can never be sure because when you're doing surveys of like a chameleon species like this, you don't always spot them. If they're in the den, what my supervisors, they were on the expedition, they would like stop the survey, stop the time, and they would go look if they could see any. And like it's very obvious when it's in the sand because they have like big piles of shells, so you can see them if it's in the sand. But if they're hiding in a crevice, I'm sure they didn't always spot them. So what I did have was abundance, which kind of can counteract this, but Maxent can't do abundance. So like it's just a lot of like with the GAM, I was able to do an abundance model and a presence absence model, but you can't always include abundance. But yeah, the absences, you're never sure. But,
I'm sorry, your answers was really accurate, really interesting, but a bit long, so we have to go ahead. But I'm sure we will ask questions for
Sorry for the other person.
It's super short, really, this question. So, with Faye and David, we go to coral reef. I take this as an opportunity to tell you, so after this session, before you board the bus, they will try to take a group picture according to the life outside. Be aware and prepared. Okay. Good luck. Enjoy.
Thank you. Good afternoon, everybody. Thank you all for coming. For those of you I haven't met yet, my name is Faye, and I'll be presenting my thesis topic on the spatial and temporal variability in coral reef carbonate production across Heron Reef, which is in the Great Barrier Reef, Australia, which I did under the supervision of Dr. Chris Roelfsema at the University of Queensland in Australia. So for those of you who are not familiar with coral reefs, I'll provide a very brief context. Coral reefs sustain over a third of all marine species globally, while only occupying around 0.2% of the ocean floor, so very substantially. Besides this biodiversity ecosystem service, they provide a range of others, such as income from tourism, food, also just simply from enjoyment, and as well as coastal protection. The Great Barrier Reef is the largest reef globally and thus holds the largest proportion of all of these ecosystem services and makes studying of it and protection of it even more important. However, like all other reefs in the world, unfortunately, they are being threatened by anthropogenic stressors associated with climate change, mainly including rising sea surface temperatures, pollution, and exploitation, all of which are projected to increase. Despite knowing this and having a lot of information about how trajectories are expected to occur, there is very little information on their resilience and potential recovery. One main reason for this is that to study them, manage them, and conserve them into the future, we need very large data sets, which span over time and space, which are very hard to acquire, as I'm sure all of you know, due to time and money constraints. Luckily, Heron Reef, which is my study site, has one of the world's longest running benthic data sets and provides a good opportunity to take a closer look into some of the parameter of reef resilience. So for the purpose of my thesis, I took the parameter of carbonate production, which I'm sure a lot of you are not familiar with, so I will keep through it. Carbonate production is part of a concept called a carbonate budget, which basically looks at reef accretion. So as long as carbonate production outweighs erosion processes, such as those from sea urchins and fish which feed on the coral, it means that the reef is accreting, it's growing, and it's a positive insight into how it is doing. The two main concepts and organisms that contribute to carbonate production are corals themselves, obviously, in growing their skeletons, but also things such as CCA, which is coralline crustose algae, and other algae such as Halomeda, which have high carbonate production values. Removal processes include, as I said, fish and urchins, but other things that impact the budget are sediment transportation, currents, anything really which can move or impact the corals and their skeletons. Only focusing on carbonate production as an indicator of reef resilience, I had two main research aims in what I wanted to achieve, which is firstly to track over time, so over 20 years of time, the changes in the benthic composition, so seeing what benthic groups are there, how do they interact, and how does this change over time, but also on a fine spatial scale. Using this data then to go into my second one, I wanted to calculate gross rates of carbonate production and see if there's any benthic composition or certain benthic parameters that influence this production, as well as possible recovery from disturbances or lack thereof. The significance of both of these aims is that this study provides the first real analysis of gross carbonate production across time and space. Carbonate production is a very widely used and accepted parameter of reef resilience, but it's only been looked at in one snapshot of time and one snapshot of space. So, having established this, I will walk through my methodology, which I split into two. First, related to my first aim, and secondly, related to my second aim. So, for benthic composition, this is my study site. This is Heron Island and the associated reef, which I have split up into geomorphic zones, so zones associated with certain geomorphic characteristics, such as depth, which being the most prominent one. Here you have the northern and southern slope, and the inner and outer reeflet. The dataset that I have access to was over 20 years annually collected and georeferenced photo quadrats, which were collected along transect within each of the zones and at random locations, pictures taken. Each picture then represents a 1 by 1 meter snapshot of the benthos attributed to each geomorphic zone. From all of this data, which is millions and millions of pictures over time, we use artificial intelligence, which in my case were the programs CoralNet and Reef Cloud, to estimate benthic composition. So to identify the benthic groups that were there, obviously firstly trained by experts and then entered into the algorithm. And from this, we eventually gain what we want, our data. So percentage cover of each of the benthic groups of interest that we will use to estimate carbonate production. For the second part, we started by looking at each of the zone and estimating their habitat area, which is not only meters squared, but we have to normalize it for rugosity, because 1 meter like this flat has a lot less surface area than 1 meter, say, like this. There's a lot more space for corals to be on and a lot more likely for them to be on. We then use that and our percentage cover data to see what the area occupied is for each benthic class. Then we go into the literature and look at widely established rates of things like linear extension, so how much it grows really, the density of each of the benthic groups, and thirdly, a conversion factor, which really normalizes for rugosity per each benthic group individually. And that will give us a calcification rate. With this then and the area occupied, we gain something called the total mass carbonate, which is a mouthful, but it eventually will help us gain the parameter that we want, which is normalized carbonate production rate in kilograms per meter squared per year. If we do not want this value only per geomorphic zone, but for the whole reef, we simply sum them all, and this is then called the gross carbonate framework production in the same units. So now, having walked through how it was done, these are some of the results that we got. So over a 20-year period, with the exceptions of 2003 and 2004 due to methodological variations, there is very clearly a much higher percentage cover in both slopes compared to the reef flats, which can be explained due to environmental factors. There's also a very, very clear dominance of a certain benthic group, mainly being branching Acropora, which is the red one here, followed by tabular Acropora, the orange, and Montipora, which is the green one below. This data can be interpreted in the following ways. So we see in years that, actually going back and showing it, we see in years that cover suddenly plummets when we go into literature and events that have happened in the reef. From 2004, so here missing, to 2008, there were outbreaks of coral disease, which evidently wipe out a lot of the benthic groups. We have then in 2015 and 2019 very heavy storms that also damaged a lot of the reefs. In 2016 and 2017, there was mild bleaching, and clearly all of these trends are very, very clearly indicating the disturbances, and we can directly see the impact of them, but also a recovery after each one of them, although being different for each benthic group. So the disturbances clearly coincide with the trends that we observe for our benthic composition. We see if you roughly estimate a recovery
the carbonate production, is that the net production or is it the gross production?
So yeah, I only looked at gross production. Normally you normalize it to a net, but then you have to account for all sorts of processes that impact it, such as currents and such, all of which was data that I did not have access to, especially over the time frame that we had. But that would be one of my first things that I would want to address going further into.
So what is it? Yours?
Gross. It's gross.
Gross.
Yes.
Okay. Isn't it for other reasons also interesting to know the mass of the standing mass of carbonate?
Very much, yes.
Is that something that you were looking into as well, or?
Yes, and actually, the lab that I worked in, they were so excited about these findings, and I'm so excited to continue working on it that will be my next step after this, to continue looking at that and hopefully expand it.
And since I'm interested in algae, how much of the production comes from algae compared to corals?
So algae are present in a much lower percentage than the coral groups and herring, just because the reef is quite pristine. However, the carbonate production rate per year is higher actually for the CCA and the Halimeda to some extent than for a lot of the coral groups, which is why the focus on these algae will be so important into the future to look at them, because they're very much understudied, especially in this aspect. So that's very interesting to consider further, especially since unfortunately recently there's been a mass bleaching event which has wiped out a lot of the coral. That was for this year, so I couldn't include it in my data set just yet. But there will be a lot higher algal cover after that with the die-off of the coral.
Thank you, Nico.
Well, thank you very much for this very nice presentation. I really enjoyed it. I think it was very well structured. It flowed really well, actually. You presented very nicely. Yeah, I missed, especially in discussion, I think all that text that you put in there could be summarized in diagrams. And it would have been much better, I think. And about the content itself, I have just 2 or 3 questions. The first one is that you used artificial intelligence, no?
Yes.
Very complex algorithms that probably include machine learning.
Yes.
Could you get a little bit more into detail of this? Do you think, at the end, my question would be, if you have the code at the beginning, and now you have the code at the end of the analysis, would you be able to interpret that?
To instruct the machine learning, you mean?
To understand what the machine did?
I would hope so, yes. Especially with the first program, so essentially from 2002 to 2018, they used a program called CoralNet, which is a lot more complex, and I definitely would not be able to understand code. They then moved on to a program called ReefCloud, which is actually made by the Australian Institute of Marine Science, so it's very much tailored towards the Great Barrier Reef, and it's a lot more intuitive, a lot more easier to understand. And the people that have developed that also work in the lab. So in some occasions, yes, we walk through it, and in some basic concepts, it's easy to understand. And especially working on it yourself, like training the images and so forth. At the end, you can look at the accuracy. So from the previous program, the accuracy was around 78%, and this one is around 89%. So it's already improved a lot, and it also has to do with the fact that it's just more easier to understand in the code that instructs it.
You're confident that at least the developers will be able to understand the final version of the code, right?
I think if you have experience in the field, and especially working with it and understand how the algorithm works in instructing it, then yes, not everything, but to a large extent, yes.
Okay. Regarding this parameter, the rugosity.
Yes.
How did you obtain that? I'm very interested in that.
Yeah.
Because it's true that it's not the same a flat surface that one that has a lot of rugosity, and that would increase the surface sometimes, no?
Yeah.
So how did you do that?
So that was very complex. I was hoping that I could collaborate with other researchers that had this rugosity data over time. But unfortunately, that wasn't the case in the end. So what I decided to do was I looked at a paper by Bozak et al. From 2015, which established a relationship between coral cover and rugosity specifically for the Great Barrier Reef. Which essentially, I took that graph, I used software to plot those points to see what the correlation was from the y to the x. And then I substituted the percentage cover of the coral that he used with my percentage cover to infer the rugosity values.
Was it a linear relation?
Yes, it was.
And what's your opinion about the future of these coral reefs? I mean, do you think that there will be a point that we will not follow any longer the same ecological succession? Because if the coral recovers, you are following the same ecological succession, and at the end, at least for some time, you have a coral reef there, then it goes back, and then it goes forward.
Yeah.
But at some point, there could be a big change. We can turn to another ecological succession that we get to algal reefs and no coral reefs. Do you think we are close to that? What's your opinion?
Thank you for that question. It's something I think about a lot. I think it depends on who you ask as a person. Me personally, I'm quite pessimistic about it, I think, which even with the reef that I've been studying, which is one of the most pristine ones you can get in the Great Barrier Reef, we see that now with the recent die-off from February, coral cover has just plummeted and it's not really recovering like we've seen previously. So I think, especially with all of the people that already presented today, the temperature estimations for what it's going to be at the end of the century, which is obviously the main threat to coral reefs and pollution and so forth, I don't think we're very far from not coming back from coral reefs, to be honest. Sorry to end on that note.
That answer, but fair enough. Okay, thank you very much.
I think we have a question.
So you mentioned in your discussion that including erosion in the carbonate budget was like a next step forward, and I just wanted to know how would you measure erosion, because it's mostly from fish, like the sampling. I don't know how.
So in the most simplest sense, what you do is you swim along the same transect that you did your photo quadrats in this case, and then you record the number and also the diversity of the paraspatian urchins that you find, mainly because they are the main contributors to erosion. It's very easy to do, so it would have been very easy to include, but just that's not something they did in this data set, so I couldn't include it.
I have another question, guys. So, in your results, when you are showing the benthic composition, so of the different groups or species with time, you pinpoint some moments or some years where we could see some collapses of notably Acropora cover. But maybe I haven't heard it, but it seems to me that you haven't talked about El Niño as a possible connection to these collapses. Is it true? What could you say about El Niño?
Yeah, that's a very good question, and that also relates a lot to the slope being more affected, because it brings in a lot more current and is more affected by currents, which would be the higher temperatures also associated with El Niño. So that would be very interesting to look at. For me, it was a lot evident to look at the localized events that happen, such as the coral disease that only happened on the island or a storm that only affected there. But that would be one of my next steps.
When only there. It was not a disease that could be linked to El Niño or the aspect. All the aspects you covered here are mostly local.
Yes. Yes. And again, that's a very interesting thing to bring up, and that would be one of my next things to look at as well.
So your work is really interesting. You're talking about Heron Island, so you're quite pessimistic for the future. Connect with a colleague just right here in this room to our students. He was telling my students, telling them, "Oh, if you want to see the nice corals, it's better you go swim." So it's true. A lot of money is still injected into coral restoration. You're talking about coral restoration?
No.
Oh, okay. So Do you know that anyway your work, or how could your work be of any use into coral restoration, notably the observation of the natural restoration of the group of species? How could your work really be of use for restoration?
Yeah, thank you for that question. A lot of coral restoration so far has focused on outplanting of corals and establishing coral nurseries and such. In my personal beliefs and also the things that I have found in this research, for instance, I wouldn't think that that's a very plausible thing to do, at least on a large scale, and not even on a reef such as Heron, where the coral cover is normally just above 80%, and you can't restore all of that with outplanted coral. So how this would be useful is to see at the benthic composition that we have, see how it recovers, and maybe highlight management for the species that increase recovery. But all of those are kind of like, you have a saying that's sweeping with the tap open, like it doesn't really help that much unless you address the causes, which is the only thing that I think this can be mostly useful for, and I hope it will be used for.
So I continue, last one from me. Could you come back on your heat map? I don't know if you call that a heat map, but yeah. Have you tried, did you check the possible significant correlation, notably the negative correlation between groups, for example, Punching, Acropora, and Fatla?
I did. Yeah, I didn't include them in my thesis in the end because it was too many results, but they were negatively correlated.
Significantly. And there is a biological meaning of that?
No, simply in the Great Barrier Reef, we know that things such as branching polytus, they are much harder to establish themselves, and once they are wiped out from a disturbance, it's very hard for them to establish themselves. So going back actually to your previous question, that would also be something very interesting to consider if you're managing a reef to improve it, for instance, to increase that diversity and to see how species also interact with each other and how that could be positive for recovery after disturbance.
Time for a really last question. Very good. Yes. Well, thank you a lot. It was a really interesting debate. Thank you. Congratulations. Let's go. David. We will continue talking about coral reef. Also about heron. Yeah. Let's use a satellite.
Yeah, started with satellite.
Right.
All right. Welcome everyone. My name is David Carrasco, and I'm going to talk about my distribution. 20 years of annual assessment of coral reef habitat, integrating field and satellite data for Heron Reef, also in Australia. It's a mouthful, but we're gonna just walk through it. This one I did also under the supervision of the Dr. Chris Rolfsen at the University of Queensland. So a little bit of background here, some ecological background. So corals, again, they don't cover much of the surface of the ocean. And even there, they're very understudied, especially their distribution and how these ecosystems are changing over time. They have historically naturally changed because of storms, diseases, and just natural competition of the habitat. But also now they're changing at a faster pace due to human-driven stressors, such as those mentioned earlier, climate change, pollution, and so on. And a lot of the times, these changes result in the overall reduction of the coral cover. So kind of what we were talking earlier, they result in algae-dominated systems. So traditionally, we monitor these changes using field-based methods, which is essentially looking more traditionally at coral cover and how I use as a standard metric. But traditionally, this only looks at particular plots and transects in a reef, so you don't really get the picture of the entire reef. And also, this is limited based on the accessibility of the habitat, because not the habitat is equally accessible and not all reefs are equally accessible. Additionally, a lot of these methods only look at after disturbance. So, for example, we know a bleaching happened. Okay, let's look what happened with the bleaching and how that affected, but we don't look how it recovers or how maybe there was a bleaching, maybe then diseases, maybe then, I don't know, bombing, fishing or something, and how the reef kind of projects over time. So this is some of the limitations that we have, and also it's hard to replicate, again, because we traditionally look at specific plots and transects, so it's hard to look at individual colonies and how they're going through time. So again, some of the limitations is upscaling. So we are not really looking at entire systems where you're looking at specific plots and transects across the reef. And also it's time consuming and expensive, which we know diving techniques, equipment is expensive. And if you want to upscale, then it just becomes more time consuming and more expensive. So this is presents a disadvantage in general, but particularly for remote habitats that are just far from shore, we just can't access or because of weather conditions, but also communities that don't have the resources to monitor via field methods. So here's where remote sensing has become a huge tool. And this is because now we're able to map entire habitats, entire reefs using object-based image analysis, which is essentially a methodology that allows us to link field data with satellite imagery. So as I mentioned, this increases the spatial resolution. So now we can look at entire reef systems and even the systems that are close to each other. And because of the availability of satellite imagery going back to 1980s, we can look also back in time how the systems have changed. So not much research has been able to really identify the ability of remote sensing to pick up these changes in composition. And this essentially is because two main factors. First, there is lack of representative field data, meaning that now we know that we have all this satellite imagery, but we can only sample the ecosystem now. We can go back in time and look how the ecosystem was. So we're not really training the satellite imagery correctly, right? Additionally, corals tend to be somewhat small, which only grow a few millimeters per year, some a few centimeters, and entire reef patches only grow a few centimeters or meters per year. And a lot of this previous research has used Sentinel and Landsat imagery, which is 10 by 10 or 30 by meter pixel resolution, which obviously is not going to tell you, unless you have a huge reef, is not going to tell you what's happening really at the finer scale of the actual coral change. So some of considerations that we've got from previous research tell us that we need to collect field data near in time to the satellite imagery so that we can actually, again, educate the satellite imagery properly and the algorithms to classify it. And we also need to collect data that is not only representative, but allows to perform calibration and validation, which I'll come back to later on. And again, that high resolution imagery is also key to be able to detect what's happening in the field at the finer scale. And the average resolution is between 0.5 to 4 meters, but yeah, so this range is usually better for detecting that change. So with this, the aim of my study was to assess the ability of remote sensing to map and detect change in the vertical position of a coral reef over the 20 years from 2002 to 2023. And the relevance of this is that, again, there is no record of such long-term and robust analysis of entire reef system. And there is no, so therefore, because we don't have this, there is no standardized methodology that can be replicated for other reef systems in the world. So starry side, surprise, Heron Island again. This reef is characterized by different geomorphic zones here, again in the Southern European reef, but this, because of the consistency of the data, considering the recommendations from previous research, we only focus on the western side of the reef, in the reef slope and the outer reef flat. And this was because of the consistency of the data. So as you can see here, we had a series of high resolution multispectral imagery between 1 to 4 meter resolution. So that falls under the range recommended from previous research. And we have an average of 3,000 field data points per year in average, right? So again, this provides what we mentioned earlier from previous research, that we need robust data that allows validation and calibration. So this one is a lot. So I'm gonna break it down here in parts. So initially we have the photo quadrats, right? So the surveys that we collect in the field, well, that they collected in the field. And using the same software, CoralNet and Reef Cloud, we classified in the different substrates representative of Heron and the Great Barrier Reef. And then we merge these classes and create mapping classes, which were mainly coral, rock substrate, and sandy substrates. And then these data sets per year are divided between calibration and validation, again, to comply with those previous recommendations. Now, the second block is the processing of the satellite imagery. So we start with the high resolution, so all those pictures that we saw earlier. And they're atmospherically corrected to get a surface reflectance so that they all give the same reflectance of the image. So you can see what you see in the field is what the image is showing. And here, using Google Earth Engine, we added physical attributes bands. So bands that that represent physical characteristics of the ecosystem, of reef ecosystem, such as slope, depth, and wave exposure. And then we also did a statistical bands that allow to improve the classification on baiting composition. And this included principal component analysis, texture measures, standard deviation, and a whole bunch of others. So once we got a pre-processed image, sounds a lot, but it's just pre-processed. We combine it with the calibration dataset, and we created via random forest classifiers, and we created benthic composition rasters. So these are essentially maps where each pixel represents the benthic composition that dominates that area. And when we had these rasters, along with the validation dataset, we run accuracy assessment because, again, you don't need to have maps if you don't know if they're good or not, so you need to evaluate how good they are. Finally, to be able to compare if this is good or not, we created trajectories of benthic types and composition over time. So essentially, this allows us to compare if the results from here are what we're actually seeing in the field. So to see if it's not like just satellites, if there's a cloud that is ruining the classification or something. So the idea was to compare the ability of remote sensing again to pick up that change. And this was looked through every year from 2002 to 2023. So some of the results, here are the maps. This is another, there's a lot of colors, but I just want you to right now focus on the classes that we map. So we map, like I said, coral, rock, and sand, but we also have a map mixed habitats like rock coral, sand coral, and mixed habitats, where neither one of the principal mapping classes were dominant. And for this, we put ecologically relevant thresholds based on some literature from the area. So now we have the maps and they tell us the composition and so on, but how good are these maps? First, before moving forward, we need to analyze how good they are. I know another one of a lot, but I'll walk you through it. So again, the accuracy assessment, it tells us how good the maps are. So when you look at accuracy, you traditionally look at three types of accuracy: the overall accuracy, producers, and users. So initially, the overall accuracy was 67%. This one just tells you in general how good the maps are. And this may sound not very high, but in more homogeneous habitats like seagrass or even land ecosystems, the typical overall accuracy lands between 70 to 80% in good confidence interval. And so here, actually, it's actually quite good because of the heterogeneity of the coral ecosystems, meaning that in one pixel, you can have many substrates, right? Like sand, dead coral, live coral in one single pixel, but this doesn't happen in other habitats. So again, this represents pretty good results for these maps. Now, the producer accuracy, this tells you how good the algorithm was at differentiating the classes, right? And here we can see that the raw coral class had the lowest accuracy of only 38%, and that makes sense because it's a mix of classes. And then the highest, this one kind of took us by surprise, is the sand coral class, which again is also a mixed class, but the coral is a lot more different from sand spectrally, so they look very different. So it makes sense that that mixture of pixels are kind of highlighted for the algorithm. Now, the user accuracy just tells you how good the maps are for the end user. So for example, if I look at a map and I put my finger on a pixel that represents coral, how likely it is that that part of the portion of the reef is actually coral in the field. So this is probably what most people usually care about because you want to know how good these maps are for the end user. So this one, the lowest was again for the rock coral class, and then the highest for the sand. It makes sense because it's the most different spectrally from the rest of the class. So now we have the maps, we see that they're good, but what do they actually mean? So here for this, we compared the trajectories of the benthic composition. And to do this, we separate the habitats in the outer reef flat and the reef slope. And this was mainly because of the difference of these geomorphic zones. The reef slope is traditionally more exposed to tidal changes, so a lot of this habitat is exposed to air in some of the portions of the reef, whereas the slope not so much. It makes sense that a lot of this habitat that is dead coral, we didn't really get to survey, so they're not so represented in the field. That where on the slope there's a lot more coral, so it's a lot easier to track. So some of the discussion here, so we see that the maps are good. So we see that we got high quality results, so the maps are actually good, and they tell us some trajectories of the benthic composition. And again, this represents a reliable and repeatable methodology that can be used in other ecosystems later on. Some sources of error that we were able to differentiate, to identify was the high level of heterogeneity. This is something we can't really control, but it's, the mixing of the substrates in the habitat really drives that lower accuracy in some of the classes. So some of these also have to do with the georeferencing error. So for example, the GPS, the points are not always fully accurate, as well as the satellite inaccuracy. And again, the spectral similarity, a lot of the habitat, like for us, looks different, but for a satellite looks the same. So that obviously drives some of the accuracy. So based on this, some of the recommendations are to plan really detailedly your field surveys. And this goes along with empirical knowledge on knowing how patchy your habitat is and so on, which also allows you to do better thresholds of the benthic composition. So for example, a 10% coral in a sand substrate doesn't affect the spectral signature of the pixel as much as 30% on rock or dead coral. Now, ecologically, this tells us that here is a resilient reef. It's resilient reef. As we saw, there was a lot of dying and then a lot of recovery, as we saw also earlier, which aligns with a lot of the GVR, the other GVR reefs, which again, it tells us that the maps are telling us a pretty good idea of what's happening in the reef. And not only this, but we were able now to look at an entire reef system rather than at single plots or transects along the reef. So some considerations here is that the habitat, these maps are obviously 2D, so we can really see, for example, different morphologies. And there's also abiotic factors that we can monitor with remote sensing, such as hydrodynamics, biochemistry, and so on. And there's also seasonal variation, that because these maps only represent a single snapshot of a year benthic composition of the habitat, but there's also algae blooms that may affect some of the benthic composition throughout the year. So this gives, again, another recommendation of the validity of combining methods. And this is essentially using these maps because they're good, they give you a good idea, but also seeing other ecological parameters such as coral recruitment, disease outbreaks, competition, predation, and so on, and looking even at different habitats and how they're connected, for example. So now to close some of the conclusions, again, we got a standardized methodology that now we can use later on in different ecosystems. And that is not only reliable, but it's also repeatable. Again, it's just we delineated the methodology that can be replicated for other systems. And this represents essentially a positive trade-off for managers because, again, we can see entire reef systems. And this is especially of importance in habitat where there is not much money or just the weather conditions don't allow for field work or where there is just no funding in general. Additionally, this also helps us to highlight the combination of methods. So again, we can see that the maps help us pick up some of that benthic composition and how it's changing, but we can also look at finer scales and understand why these things are changing, right? So we can look at the coral ecology again, and some associated biotic and abiotic factors, so we can look at carbonate budgets, predation, and all these things that can help us understand more why the satellites are picking up what they're picking up. And this also lays the ground for future research, where we essentially need to implement this at a larger scale. So not only look at a single reef, but also reefs that are connected or entire reef systems, such as the Great Barrier Reef or the Mesoamerican Reef or so on. And then we can also look at drivers of change, because with satellite imagery, you can look at, for example, sea surface temperature. You can use chlorophyll A sometimes to some extent as a proxy of pollution, of nitrogen pollution, to some extent. So you can start looking at not only how they're changing, but what's driving these changes. Also further, we can start looking at coral bleaching because it's something that is very understudied, especially with remote sensing. So we can start looking at how, for example, before, during, and after the reef has behaved and has changed. And with this, I say thank you. Here are some of my references for those who are interested, and any questions.
So here we go. Who would like to start? Cool.
Yeah, I can start with this. Sure. I don't have so many questions.
Okay.
Yeah. Thanks for a nice presentation. You did a lot of work with the method. Yeah. But now in the end, you show us really nice maps. And then you showed us some trajectories, but that was like a mean value for a larger area. But if you look into the maps, you must have been looking into the maps. Can you tell me anything about the dynamics in, like, the spatial dynamics?
What do you mean by spatial? Like within the reef?
If you go to one point in the map several years.
Oh,
yeah.
Yes.
Yeah. How can you give me some indication of the? Is it always the same thing?
Yes. Okay.
Or is it a little bit more dynamic? Because this could be really interesting to say that normal coral reef could be really, really stable, but maybe it's more dynamic than we thought of.
Yeah, no, and that's a really good question, because that's actually one step that we're going to do further in order to publish this. Is we're going to run a sensitivity analysis where we're going to see, for example, because we see, I don't know if you can see from there, but a lot of this slope, so especially this outer range of the reef, it remains mostly blue, right? So meaning mostly coral throughout. And I know it's kind of hard to see, but I hope you believe me there. So it's there. But in the outer reef flat now, and this makes sense also biologically because it's a very fragmented habitat, it's really hard to really pinpoint one single point that is always correct because we also have the GPS error. So for example, if I put in the field, if I collect a point here that is coral, but the GPS puts it over here, if you know a coral reef, that could already be dead coral or sand, right? So it's very different. So it's hard to know if, for example, in that fragmented area, if that very decomposition is correct, right? And that goes back with accuracy assessment. So that's one of the biggest challenges, especially again in those fragmented areas where some of the habitat, a lot of the habitat is not accessible because the slope is characterized by a meter, by depth of 5 to 10 meters, but the outer reef that sometimes is exposed to air, sometimes it can be 5-meter depth. So a lot of the habitat is not always accessible and being able to be represented in the field data. So the sensitivity analysis hopefully is going to tell us how dynamic these habitats are, but also keeping in mind that we need to have still reliable maps and reliable results. If that,
yeah, I was thinking about if you have been looking a lot to these maps.
Yeah.
If you just can give me some, well,
yeah, I think
love doing the sensitivity analysis.
Yeah, I think the biggest point to the biggest thing to point out is that obviously the slope remains mostly coral, and that also makes sense ecologically. But here in the reef, in the other reef flat, sometimes some pixels go from coral to sand and then to coral again, which ecologically doesn't make sense because a coral can disappear and then reappear in the sand 2 years later, right? So this is some of those things that hopefully the sensitivity analysis will tell, especially at the different geomorphic zones.
Thank you.
Maybe just on a follow-up on that. So do you have a variation of the benthic type with time? Like not for one, it's really hard to have one pixel at a time, but this is here. So
yeah,
here you see a dynamic.
Yeah.
Is there a possibility to test it in one way or another?
That's what, again, that sensitive analysis is hopefully going to tell us, because as you can see here, the field data is quite, I mean, it's not super smooth, but it's quite smooth, right? But you can see there was a decline and then a slow recovery, which again, ecologically makes sense. But then the satellites, you see somewhat of like that, but then you see these kind of spikes, right? So hopefully that sensitivity analysis is going to tell us how misplaced this point could be. So for example, instead of being here, it's going to be here, and then that's going to kind of smooth out that transition. So hopefully, and that's, especially in these areas, is where we're hoping to see more of that smoothing of the lines of the trajectory of the benthic composition.
Yeah, well, congratulations for the presentation. I really enjoyed it. I think you made an amazing work explaining to us all the different parts of your work. Although I think you rush a little bit too much, and that you were speaking too fast.
It's a lot of information.
Yeah, it's a lot of information. Yeah, I know. And about the content of your presentation itself, could you tell us a little bit more about the satellite itself? Because it's amazing what this one can do.
Yeah, so that's actually one really good question, and I think you asked that because, like I said, most research usually uses Landsat and Sentinel, right? They're huge pixels where you can see in some places like entire like communities and all this, right? But these are at 1 to 4 millimeters, and we have actually a lot of different meters. And here we have different satellites as well. So there's also that sensor error that is not the same across.
It's a merged product of different.
Yes, it's different satellites. So as you can see here, I should have mentioned that, and you're correct. We have a QuickBird, we have Cassie, we have WorldView 2 and 3 Planet. So it's a series of different satellites. That's why I mentioned that the resolution is between 1 to 4 meters, because they're not all the same.
So, okay. Why is that? Because I guess you could have at least one. Why are you using these different types?
Because before 2016, there was no Planet data, which is free. Yeah. Before you had to pay. And if you know love satellites, the data is expensive, not always available. Yeah. And a lot of times you can see here this patch of cloud. Very locally, there was only one patch of cloud, because a lot of times the cloud is covering the whole reef. So even if you try to go back in the archives, a lot of these satellites, they're fully covered by a cloud. So this is something very hard to deal with.
You mentioned something important there. It's not free access, right? It's expensive.
Planet is free to some extent.
Planet is free.
Yeah, but the rest is very expensive.
Okay. And you don't have data there in 2003?
No, there was no funding that year. So yeah. No fun in the okay. And what environmental variables do you think that could affect the performance of these satellites? Because what about depth? Yeah. Because it's not the same that you have your coral at 1 meter depth at 3 meters. Yeah. The spectral pattern of that image is going to be different depending on the amount of water that is on top of the coral. Yeah. So, did you account for that, or did the product account for that in some way? Yeah, we did. So initially, okay. So a lot of research, what they do is they do depth correction, which is essentially looking how the refraction and the reflection of the bands when they come back and is picked up by the satellite. So I know it's a lot of words, but in the physical attributes band that I mentioned here, we had a depth, slope, and waves, which are three of the most characteristic physical attributes of coral reefs, because it changes the spectral signature. So obviously, when you run the random forest classifier, it takes into consideration those bands. And here in the statistical bands, we also had like ratio bands of depth and slope, or like the bands, because for example, the red band doesn't penetrate more than 5-10 meters, good, but then after it gets refracted and reflected. So it starts messing up. And so we clipped these maps. As you can see here, I have depth water over 10 meters. So because that's where mostly the red band, for example, doesn't penetrate any further. So we click there because that visibility is threshold there. Okay, yeah. I see. And just a little question on your time series, the ones that you were showing before. These ones. Yeah, you see some correspondence in between field data and satellite imagery, but why not correlating them? Like, why not overlapping them? And correlating them, yeah, well, one versus the other. Yeah, that's again kind of what I was saying earlier. We're going to try to do that sensitivity analysis first to kind of smooth out those transitions, because these peaks ecologically don't make sense, right? Like you can have this is like 10% sand increase kind of randomly. It's a bit too much ecologically. So hopefully with that sensitivity analysis, and we understand where these points can be displaced, then we can correlate to this, because this is what we know we're seeing in the field. Right. So you're going to do some smoothing on the satellite? We're going to hope to be able to run a sensitivity analysis again to see what's driving those peaks, random peaks, so that then we can correlate to the field data. Okay, thank you very much.
Hey, whoa. Yeah, so you started in 2002. Is there a reason? I guess yes, but why you could not go further in the past?
Yeah, so this is when the supervisor started working there, and that's when he started running the program. Before that, there is data, but there is no field data, so we can't really go back in time and collect the data. So this is when they started running this yearly monitoring effort.
It's not in 2002, not in 2003. Okay.
Yeah,
you want to go large, I understand. You want to extend that point of view to the barrier level, for example. But if we stay at the Heron scale, would it be possible to imagine a drone sampling that would make it much more accurate locally? Yeah, for sure. Drones can have resolution of 40 centimeters, 30 centimeters, depending on the sensor. So hopefully you can, but then you start thinking, but then you have to start thinking of differences of the sensors, because satellites in general are very similar, but now with drones you're going to have a lot of different variabilities in the collection of the data, because the sensors are different, because a lot of them just use normal cameras, right? Whereas the satellites have specialized equipment. So you can, and it's hopefully something that will continue, we'll do later on, but just for consistency, satellites is... No, anyway, it could be in addition to satellites. Yeah. Hey, it's your turn. You're shy? No, finally, I'm happy you can ask a question.
Can you tell me a bit about the image selection process for each year? Like, do you pick one representative image, or you mosaic across the year? Is there like seasonality that you're counting?
Yeah, so a lot of these are singular snapshots of the year. So for example, I just don't have it here, but in my document I have a table of what date, particularly each image was collected and how far it was from the field data. So you can see that obviously some are going to be like the same day or within the same week, but some others are a few months apart. But the images, they're all single images, so we didn't stack them. Because if one image is expensive, now imagine many, right? To stack is a lot to process, and also it started becoming too heavy and it doesn't process. Because for example, Google Earth Engine, it has a very limited memory. So when you start putting high resolution imagery and you start doing all these like statistical bands and physical attributes, start getting too heavy. So for like even some of these images took hours to load to get a map. So that's why we didn't stack them.
Congratulations. Ali, go ahead. Last question.
I have one question. So I know that Heron Island shouldn't have too much variability in this, like for a season. For example, if one image was from like January and one was from September, would there be any like big difference? Could imagine a big difference?
In that, it depends. In some it does, but that's mostly in this area that we didn't map, the lagoons. Because there is not much field data here, so that's why also we only focus on this area. But this habitat is, for those who can't see this habitat, this habitat, there is a lot of algae blooms during the summer. So that habitat does get a lot of algae blooms, so there is a lot of competition with other algae for coral and so on. So again, that's something that we didn't take into consideration for the results and why we excluded habitats like that. Yeah.
Before we break all the microphones of the room, I think. Thank you for your questions and congratulations again for before you